Let us see after 15th day after hatching. It is 23 mm in length, anal fin with 2 to 5 rays, caudal rays 36, basal 2 by 3 of the dorsal fin pigmented, black chromatophore band in caudal region, posteriorly prolonged from the center passing through the origin of caudal rays and dividing the fin into two. 18th day after hatching, how it looks. It is 25 mm in length. Body is dirty yellow above lateral line and yellowish white below it. Scales present but not clear near the caudal peduncle and in the abdominal region. Black chromatophores are on the anterior margin of the dorsal fin in the form of dotted line. Ventral fin is with 9 rays, 32 caudal rays. Two pigmented grayish crescents, one on each half of the caudal fin lobes behind the dark triangular areas are on the caudal peduncle. Now let's see how it looks after 20th day after hatching. It is 26 mm in length. Body is golden colored. Scales are prominent and fully covering the body. Two dark bands, one above and one below the lateral line, are along the body up to the caudal region. Dorsal fin is with 3 to 13 rays. Caudal rays are 34 and rays are covered with orange pigment dots. A black band along the entire width of the caudal peduncle, concave posteriorly, and are irregular in shape along the anterior margin. Behind this band, two faint crescentic areas separated by two colorless streaks. Now, after 25th day, it is 30 mm in length. Body is golden color with coppery reflection from operculum. Barbels are prominent. Upper margin of eye is faintly orange. Dark band in caudal peduncle is quite prominent. Upper lobe of caudal fin larger and pointed compared to somewhat round edge of smaller lower lobe. Let us see the life history of other cultivable fishes. Of the exotic food fishes, these are more important from the point of fish culture. One is common carp, silver carp, grass carp, and tilapia. Let us discuss about one by one. Now, common carp. What is the zoological name of that? Cyprinus carpio. Now, the fertilized eggs of common carp are small, spherical, demersal, and adhesive, measuring from 1 to 2 millimeter. The perivitellin space surrounding the egg is narrow. Yolk is usually yellow to light brown in color. Embryos hatch out within 2 days to 7 days, depending on the water temperature. The newly hatched larva is 4 to 5 mm in length and has a prominent yolk mass. The newly hatched out larva adheres to the leaves of aquatic plants by means of semen glands and remains in this condition until the yolk is partially absorbed. The yolk is absorbed within 2 to 6 days, depending on water temperature, after which the post larva, measuring 6 to 7 mm, start normal movement and commence taking nourishment from the environment. If suitable food is available, the growth is rapid and fins get differentiated. Within 12 to 15 days of yolk absorption, all fins develop the full complement of race. This is a common carp and these are the larval stage. It shows the growth of the larva, how it looks in each stage. Next, coming to silver carp. Freely oozing viable ova of silver carp are pale bluish in color. Fertilized eggs are demersal and round and ashy gray in color. Within 5 minutes of fertilization, eggs start swelling, and during the next half an hour, the process of water hardening is completed. The full swollen fertilized eggs measure 4.2 to 4.7 mm in diameter. The length of the newly hatched larva of silver carp is 5 to 5.5 mm. The length of yolk sac is 3.33 mm. Height of the body is 1.25 millimeters. Height of the yolk sac is 1.09 millimeter. With 42 to 44 in number of myotomes, of which 14 to 16 are located behind the anal level. Yolk sac are fully absorbed by the end of the second day of hatching, and thereafter spawns start feeding from the environment. They measure 7.4 to 7.53 mm in total length and 1.21 mm in height of body. The silverfish eggs, just now we were discussing, they look bluish in color, and the fish looks like this. Next, coming to grass carp. This fish lays eggs in stages. Eggs are pelagic and undergo development as they float downstream. Ovarian eggs are of yellow and deep golden brown in color. Fertilized eggs get fully swollen in half an hour and measure about 4.5 mm in diameter. Newly hatched larvae measure from 4.5 to 6 millimeter in length. Length of yolk sac is 2.84 millimeter. So maximum height of the body is 2.9 mm and maximum height of yolk sac is 0.78 millimeter. Yolk mass is conspicuous and of a pale brown color. Of the 45 myotomes, 14 are posterior to 37 at the anal level. 2-day-old post larva, just after absorption of yolk sac, is about 6.98 millimeter in total length and 0.9899 mm in maximum height of the body. The, the fertilized egg looks like this, and this will be the maximum. This can grow bigger than this, the grass carp. Now let us see the life history of morels. Fertilized egg is round, non-adhesive, buoyant, free, and straw yellow in color. The yolk is capped by a distinct blastodisc and contains an oil globule liable to disintegration to smaller 5 to 6 globules, unequal in size. Perivitelline space is of medium width. Diameter of the egg is 1.2 mm and that of the yolk is 0.9 mm. Now, how it looks after 10 hours after fertilization. Anteroposterior axis is distinguishable. Cephalic portion being broader. Forebrain region discernible. Invasion of the yolk is complete. After 14 hours after fertilization, the embryo with six somites is embedded all over its length in the yolk mass. Optic cups are clear. Height of the embryo is 1 mm. How, how does mother looks after 16 hours after fertilization? Mesodermal 9 somites are present. Notochord is more discrete, and the four mid and the hindbrain regions are also defined. Cephalic portion is broadened. 17 hours after fertilization, how it looks. The somites range from 13 to 15 in number. Ectodermal thickening to form lens of the eye is indicated. Tip of the tail is from the yolk at the last somite. Embryonic fin fold appears. Cuptor's vesicle is visible. Cephalic region is depressed. Height of embryo is 1.1 millimeter. 20 hours after fertilization, how it looks. Somites are 22. Embryonic fin fold on the ventral side extends up to the 10th somite. The lens is now fully formed in the eye. Olfactory placode indicated. Cuptor's vesicle gets diminished. Blood circulation can be seen over to yolk into the rudimentary heart lying anterior to the yolk sac. The heart beats, it beats very rapidly. Some dendritic melanophores appear over the yolk. 22 hours after fertilization. Somites are 24 in number. The embryo encircles the yolk and covers practically the entire capsule. Tail is free. Olfactory pits and auditory vesicles are now well pronounced. Some melanophores appear above the neural cord, over the trunk and caudal regions. Heartbeat is very rapid. Cuptor's vesicle disappears now. How it looks after fertilization after 24 hours. Twitching movement of the embryo is vigorous and lashes the tail vigorously against the capsule, thereby springing up the head which suppresses the wall and brings the embryo out. The newly hatching larva is dull brown in color, 2.7 mm long and 1.1 mm height. The fin fold originates dorsally at the second myotome, runs ventrally, the region of the vent, and goes up to the yolk sac. Third and fourth ventricles of the brain are then prominent. 8 dendritic melanophores appear at the caudal region. Linear melanophores appear at the caudal region on the lateral side above the body. Now, 3 hours after hatching. Its length is 3.1 mm and height is 1.4 mm. The heart is two-chambered. Circulation can be seen around the notochord, in addition to the brain and yolk. Blood corpuscles are reddish yellow, showing formation of hemoglobin. The anal opening is marked by a slight invagination. Now, how it looks 8 hours after hatching. Its length is 3.5 mm now and height is 1.1. Myotome count is 31. Bulge of the yolk is reduced. Some more melanophores appear on the ventral side of the notochord and the dorsal side of the body. Circulation is conspicuous at the optic region. Some pigments are interconnected along the intermyoformal septa. Now let us study about the morels. See how they look? 15 hours after hatching, its length is 4.2 mm and height is 1 mm. Auditory capsule moves closer to the eye. Pigments over the iris are now denser. Mesenteron is seen as a blind tube and nephridial duct are conspicuous. Location of pectoral fin is marked by a thickened conical patch. 24 hours after hatching, its length is 4.3 mm and height 10 mm. Myotome count is 32. Buccal imagination appeared. Pectoral bud is now well pronounced. The air sac is differentiated as small tube below the pectoral fin bud. Eyes are fully pigmented. 36 hours after hatching. Its length is 4.4 mm. The pectoral fin is a round membranous flap and is being actively used for free movement. Mouth is formed as terminal opening. Vent is formed. Rudimentary gill opening and nasal plates differentiated. A thick band of melanophores run from the post-orbital region to base of the pectoral. 2 days after hatching, its length is 4.6 millimeters. The pectoral fin is paddle-shaped with undulating dorsal margin. Larvae move horizontally in schools. Lower jaw is well developed. Trunk region is a little constricted at the nape. Only a vestige of the yolk is seen. Let us see now 18 days after hatching. Its length is 6.3 mm. Dorsal contour of the head is convex. Basal thickenings appear in the dorsal and ventral fin folds. Light caudal rays can be distinguished. The middle ones showing articulation, the caudal fin is dark along the margin with bright yellow blotch in the middle. Silver and greenish pigments appear on the orbital rim and also on the post-orbital region. Now how it looks 20 days after hatching. Its length is 7.2 mm. The fry assumes nearly the adult characteristics except the color pattern. Rays start forming in all the fins from the 18th day onwards. There are 14 rays in caudal, 19 in anal, 25 in dorsal, 17 in pectoral at this stage. The fry has characteristic iridescent lemon yellow and dull longitudinal blotch bands. Aerial respiration is noticed. The fry move actively in schools. The ventral fins are differentiated on the 23rd day when the fry measure 14 mm. Scales appear on the body by the 26th day when they are 18 mm long. Let us see now the life history of prawns, cultivable prawns. Generally, the pineared eggs hatch and metamorphose through four distinct stages. They are Nauplius, Zoea, Mysis, and the post-larva stage. Now let us see the prawn Peneus indicus. This species prefers higher salinity. The post-larva range in size from 5 to 10 millimeter in length. Post larvae are transparent white in color, excepting for pink coloration at the tip of the rostrum and the tip of the telson. Antennular peduncle tipped by the light reddish coloration, light yellowish color on eye stalk. A few branched brownish chromatophores tinged with yellow are present on the mid carapace and on few of the mid abdominal segments and telson. A single dorsal tooth is present on the rostrum. The rostrum extends slightly beyond the eye in the first post larvae. A well developed supraorbital hepatic spine is present. A median dorsal and lateral spine is present on the fifth and sixth abdominal segments. Anal spine is also present. Post larvae swim fast, moving along the edges and surface. If you look at the prawn, the eye is prominent. This is the eye stalk, rostrum, the stalk-like structure above the head, and this is the carapace. It covers the thoracic area. And these are the segments present in the abdomen. These are called as abdominal segments. And the tail portion, it is called as telson. And the coverings are called as the uropods. These are the legs of the prawn, swimming legs. These are called the swimming legs. These are called as pleopods. And these are the walking legs, periopods. So walking legs are called as periopods and swimming legs are called as pleopods. And we see the prawns have long antennae. It is, students, it's very important to learn the labeling, learn the parts of a prawn. Now, if you see the life cycle of a prawn, the different stages, the mature adult, the prawn, it lays eggs. And once they lay eggs, the initial stage is called as nauplius. This nauplius grows into zoe. And then it grows into mysis. Then it grows into post-larval stage. And then it transforms into juvenile prawn, the younger one. The juvenile prawn matures into adult prawn. These are the larval stages of prawn. The different larval stages just now we were discussing. And we can see a number of varieties of prawns, how they look. Some are orangish in color, some are red in color, some are grayish in color, some are white in color. The body structure is different. Some they have the striations also, like stripe, orange and white stripes on its body. Some body is white in color and the segments are orangish in color. These are various species of prawns. Let us discuss about the different stages of the larval stages. The late mysis measures 3 to 3.5 mm in length. The size range of post larvae is 3.75 to 4 mm. Rostrum is very small, falling short of the middle of the eye stalk. The tip of the rostrum is acutely pointed. Two principal teeth are born at the base of rostrum and one smaller tooth in front of the first principal one. The dorsal spine present on sixth abdominal segment only. Post larvae have characteristic brown color. Reddish Pigmentation on the ventral side of abdomen, present on entire telson. In later post-larvae, on the sides of the carapace, bluish-brown spots concentrate to form a band. A M-shaped bluish-black patch is also seen on the dorsal side of abdomen at this stage. Metapeneus brevicornis. This is another species. See, the size range of the post larva is 3 to 3.5 mm. Rostrum is short. It just touches the border of carapace. It is brown in color. In juveniles, rostrum shows only slight increase in length, exceeding the tips of the eyes, while the basal elevation is more pronounced. Dorsal teeth are seen 7 in number. Rostral length is 2.2 millimeter. Deep yellow coloration is on carapace. Black red bands along the lateroventral aspect of abdomen. Red spots are present on the uropods and telson. Now let us see the summary. We had discussed about the life history of rohu, marils, and prawns. Rohu attains maturity at the end of second year in its life. It spawns only once in a year. Eggs of rohu are round, transparent, non-adhesive, and red in color. Yolk spear is without oil globule. Newly hatched rohu larva has transparent, laterally compressed body and is characterized by the presence of gill slits, pectoral fin, and median fin fold. Larva of rohu takes 20 to 25 days to get all adult features. The fertilized egg of maril is round, non-adhesive, buoyant, and straw yellow in color. 6 somites are seen in the embryo of maril after 14 hours of fertilization. Common exotic carps are silver carp, common carp, and grass carp. In marils, scales appear on the body by 26th day of hatching. Common cultivable prawns are Peneus indicus, Metapeneus monoceros, and Metapeneus breviornis. The larval stage of prawn are Nauplius, Zoe, Mysis, and post-larval stages. Now, from this chapter, what are the questions you can expect are write about life history of Rohu. Then you need to explain the entire days after hatching, the hatching period. Everything you have to, what are the slides I had shown and explained, everything you can mention. So you need to write up to 30 lines. Or write about life history of other cultivable fishes. Write about life history of maril. Write about life history of cultivable prawns. These questions are repeatedly asked. You can write up about 30 lines each. Next, these are the some of the questions which you can answer in 10 lines. Write about Peneus indicus. the life history or the life cycle of Penis indicus, life cycle of Metapenis monoceros, life cycle of Metapenis brevicornis, or life cycle of silver carp, grass carp, and common carp. So these are the slides where the entire information is there. Life history of Rohu. Then this is the life cycle of a fish, Rohu fish. So how it looks after 30 minutes after fertilization. Then how the cells divide. Then it's called cleavage. How the next is almost 1,000 cells are there. Then the shield stage, the shield is grown. Then a gastrula is the group of cells. Gastrulation and epiboly. Then organogenesis, organs start forming. Then it is hatched into a freely swimming larva. This larva, this grows into an adult. Then the characteristic features of Rohu after 24 hours, after 36 hours of hatching, how they look, after 48 hours of hatching, 72 hours of hatching, then days, after 3 days, after 4 days, how they look, after 6 days and 7th days, 8th day, 12th day after hatching, 15th day, 18th day after hatching, then 20th day, 25th day after hatching. So these are the stages you need to explain when the question is asked about explain the life history of Rohu fish. Then these are the other fishes, like common carp, silver carp, grass carp, tilapia. About common carp also you can explain how the larva looks, how the fish looks. Then the silver carp, the size of the egg, how the growth takes place, how the eggs look bluish in color, the silver, the it when we see we can understand how it shines like silver. And then the grass carp, what is the length of it, how the eggs look like, how big it can grow. And then life history of murrels, 10 hours after fertilization, 14 hours after fertilization, 16 hours, 17 hours, and 20 hours after fertilization. You can make a table of the growth. So how the growth is increasing hours by then 22 hours after fertilization, 24 hours after fertilization. Then it comes to 3 hours after hatching, 8 hours after hatching. Then how this is about murrels, how they look, the hatching period, how 15 hours, 24 hours after hatching, 36, then coming to 2 days after hatching, 18 days after hatching, 20 days after hatching. Then the life history of cultivable prawns. These are the 4 stages, and this is very important. The parts of the prawn, you can practice the diagram and you should understand the labeling of the prawn. And these are the different stages of the life history of a prawn, where a mature adult lays eggs. Once it is the starting stage of the larva, it is called as nauplius. Then it changes into zoe. Then it changes into mysis. Then this is the larval stage, which changes into a juvenile, that is the young prawn. The juvenile matures into an adult prawn. The same larval stages of prawns. These are the different species of prawns, and these are the different stages. How Metapenis brevicornis looks like, and that's all. So hope you have understood the chapter, students. Thank you.